Assalamu alaikum, sir.
Wa alaikum salam wa rahmatullahi wa barakatuh.
Assalamu alaikum wa rahmatullahi wa barakatuh.
Wa alaikum assalam wa rahmatullahi wa barakatuh. How you guys are doing? Hope everything is fine.
Alhamdulillah. Alhamdulillah.
MashaAllah, mashallah. Just give me 1 second. Inshallah, we can start. We discuss about enzyme properties. What exactly are enzymes? I think you are very much acquainted now with the concept, basic concept. Last time we were discussing different factors that could affect the enzyme reaction. You know, the speed of reaction and the activity of enzymes. So 2 major components we discussed, 2 major factors. Who is going to tell us those factors? And how would it affect the enzyme reaction? Yes, Hania?
Teacher, it is, they are all proteins.
Okay, enzymes are protein in nature. So what my question is that we discussed some factors that would affect rate of reaction by enzyme. So what are those 2 factors that we discussed last time, if you remember?
Can I tell one?
Yeah, go ahead.
The temperature, because if the temperature increases, then the enzyme works faster.
Yeah, first of all, temperature itself, yes. If temperature is increased, there is optimal temperature, of course, and if you're increasing further beyond that limit, each enzyme has got some stability at certain temperature. If you are increasing further, you might damage the enzyme, which is called denaturation or denaturing the enzyme. Okay, yes, that's good. What is another factor? Yes, Hania? Okay, any other factors somebody would like to focus on? Sorry?
Can I tell?
Yeah, go ahead, please.
pH.
pH, yes. pH of the environment where the enzyme is working, right? So anything you want to relate with the pH? Any example in your mind? What kind of pH? What do you mean by pH? And if you have any example in your mind?
pH is like how much an enzyme is acidic or how much it's alkaline.
Exactly. So what is an acidic pH and what is an alkaline pH? Yes. So remember we discussed a point where there is a pH, it's a neutral point, and then you have acidic pH, and then you have alkaline pH, alkaline environment or basic environment. We remember some of these values. Who's going to tell what is the neutral value? Yeah, any one of you. Yes, I can see 2 hand raised. So anyone could participate. Go ahead.
7 is neutral.
Neutral is 7. Yes, very good.
Under 7 is acidic and over is alkaline.
Very good. Yes. So pH will also affect. Yes, Sabiha?
1 is the most acidic and 14 is the most alkaline pH.
So below 7, yes, of course, is acidic. So if it is closer to neutral point, so that is weaker acidic. That environment is weak acidic, right? So if it is away, like 1, 2, up to 3, for example, stronger acidic. But if you want to compare 6, for example, pH 6 is closer to the neutral point. So it's going toward neutral and then basic, okay? Yeah, that's true. Very good. So and I think pH is also affecting the enzyme, right? Because some enzyme, there's what we mentioned in the intestine, for example. Which environment is an intestine? Is it alkaline or acidic? Yes.
Acidic.
Acidic. Very good. So no, intestine is acidic or basic? Intestine is basic, right? We discussed before some of the proteases that are active in basic environment. Your stomach has got acidic environment. Okay, some of the enzymes are there in acidic environment. They are active. Some of the trypsin, for example, proteolytic enzyme, but proteases are also effective If you remember, we were talking about digestion of proteins. So we mentioned that an intestine is a very good environment because enzyme will be very active. Okay, if it doesn't favor the environment for enzyme activity, so enzymes sometimes are also destroyed in that environment. They are not stable and they cannot work. The active site may not remain the same. So that's why it's not going to work on substrate. Okay, so let's talk about some of the key points in the summary. So they are saying that enzymes speed up chemical reaction. That is one of the property that we discuss. Enzymes are needed. They are constantly working in our body, especially in our cells. So you agree with that? That will speed up the reaction, right? So anything that is speeding up the reaction, what do we call it? What is mentioned? What the term they have mentioned in the chapter? So if something is speeding up the reaction, what do you call it? So enzymes are also called?
And they call like catalysts and the catalysts. And
Yeah, they are catalyst. Catalyst is enzyme, separately enzyme, right? Catalyst. It is working on.
enzymes are called the biological catalysts.
Yeah, very good. Enzymes are biological catalysts. Anything that are increasing the reaction of the chemical reaction in our body, in our cells, and that's why they are biological catalysts. The another point that we discussed was here the in the summary. Okay, in the summary part, you have breaker enzyme that split up small molecule, large molecules into smaller ones, and then you have builder enzyme. So they are just focusing on key. Okay, you may remember some of these example, and you go through the chapter. You have all builders enzyme. In building mean they are bringing together small molecules. Okay, and this is how they are making uh, bigger molecules. They are builders, and breakers are the one that will break down bigger molecule, carbohydrates, starch, fats, for example, proteins, and all the stuff. So you cannot use these essential elements without breaking them down into smaller unit like amino acids, glucose, and fatty acids that can be easily absorbed. Okay, and enzymes are very selective. Okay, so I think these concepts are already discussed here in the question. So let's go to question. So who is going to read question 1? That okay, 3 participant. Let me see. uh, okay, okay, Sabiha.
Should I just read it or should I also give the answers?
Can we can't hear you properly. I think it's better if you are not using your headphone. It's better to use your built-in speaker in the laptop or your device.
Sir, I'm saying, should I just read it or should I also tell the answers?
I think, yeah, part of it you could answer. For example, A, this is for you, A.
Okay.
Read it and answer also as well. And you explain it. Why? What do you understand? Why did you choose that particular option?
All right. Enzymes speed up the rate of chemical reactions.
Yes, enzymes speed up the rate of chemical reaction. That is why they are called catalysts. Very good. Okay, let's go to, you can also read part B because there are so many parts, so other students can also, yeah, go ahead, part B.
Enzymes are specific because they only work on one substrate.
Yeah, they are very specific. They are very selective also, right? Enzymes are very selective, very specific in their action because they are working on one substrate. So would you like to give some example of substrate and enzyme, which is very selective for a particular substrate?
Protease only works on proteins and lipase only works on fats.
Right, lipase are breaking down fats while proteases are breaking down proteins. Okay. Part C was going to read part C. Next student.
Can I read?
Yes, okay.
The substrate fits into the active site on the surface of the enzymes.
Very good. The substrate fits into the active site on the surface of the enzyme. Okay, that's very good. Excellent. Yes, you know, active site is that particular, the whole part of enzyme is not active, right? Only specific area in the enzyme. And if you're called, what is that area? You know it very well now. That is an active site of enzyme where substrate can bind, okay? Enzyme can easily attach substrate there and then is free. Okay, the enzyme will work on that substrate, either breaking down or bringing together. So depending on nature. Okay, go ahead to next part. Okay, who is going to read that? Rafe?
I just read the C.
Sorry,
I read C1.
number D. D, yes.
I read C.
Sorry, we can't hear you. Could you please speak loudly?
I'm saying that I read the part C.
You already read the part C. Okay, great. Okay, Omahel, would you like to read part D? Yes, Omahel? You can hear us? Oh, Omahel, we can't hear you. Okay, let's see. Aisha, would you like to read part D?
With an increase in temperature, the rate of reaction increases, but eventually a temperature is reached which denatures the enzyme.
Exactly. So this is how temperature is related, right, to the activity of enzyme. Very true. Very nice. Yeah, this is a very, very important factor that you should keep in mind, that enzyme will gradually, the activity of enzyme may be increased if you are increasing temperature up to a certain extent. But if you go beyond that limit, then enzyme will be denatured. Okay, who is going to tell us? Sabia, would you like to answer why increasing in temperature would increase rate of reaction by enzyme?
The increase of temperature would increase the work of enzymes. It would like be faster.
Like, yeah, this is what we mean by increasing reaction, mean they are faster, but why they will be faster in higher temperature and they will be slow in lower temperature? Oh, anybody would like Hania?
The high can I Sabiha?
Okay, Sabia. Okay, go ahead.
The higher temperature, like the enzyme, the enzyme, they will work faster. Like when if you heat up water, the molecules will speed up. Just like this, the enzymes will also like speed up their the reactions.
Very good. So when you are increasing temperature, initially you're already increasing the thermal energy, right? So enzymes are also molecules, substrate. The interaction or collision of substrate with enzyme is increased when increase in temperature, right? So that is why we would say that temperature will speed up the reaction. Very good, yes. But if up to that's, I mean, if you remember, we discussed before that up to certain limit, the active sites are kind of vibration. If they are vibrating, they are moving faster. But up to certain limit, when you are increasing temperature too much beyond the capacity of enzyme, so it will vibrate so much that the active site will be damaged. So if the active site is not resembling to fit the substrate, the interaction with substrate or binding of enzyme will not take place at a very high temperature, and we call it denaturation. Denaturing is an irreversible process, so you cannot make this enzyme active again because you denature, already destroyed. Okay, good. So E, who is going to read E? Those who have not, Aisha. Yes, Aisha. Part E.
Enzymes can be reused, so only small amounts are needed.
Yeah, they are. So who is going to explain? Rafi, would you like to explain? Why do we need small amount of enzyme? Why not? If you have bigger amount or larger concentration of enzyme,
We
would it still
need
be beneficial?
okay. We need small amount because the once the substrate when the substrate leaves, that's it. The active site can be used again. So new substrate can come and join with the active site. Like it will go on like this when all the substrates uh, are made.
Yes, okay. So that is why we need smaller amount of enzyme, because reused, once the substrate attached to the active site of enzyme, so enzyme is already converted that substrate into a product, right? So enzyme is free now. We need smaller concentration. Muhammad, do you have some comment? You want to add something more? If you are increasing concentration of enzyme, do you think would it be beneficial? You would have more product?
No, sir.
Okay, why?
So because the less amount of enzymes can also do the same work, so we don't really need so much enzymes.
But if you are increasing the concentration, do you think it would also increase the rate of reaction, or you could say the conversion or uh, yield or production will also be increased by the enzyme?
No sir, no sir.
Okay, you remember the reaction might be faster if you remember a catalyst that we are using for the potatoes, right? So
Yes
how
sir.
it is converting glucose into starch, and if you break down or chop the potato into small pieces, so you probably have more enzyme activity. So concentration, definitely you don't need it. Smaller amount is required, of course, but if you are increasing concentration, it might increase reaction product. You know, if you are basically increasing chances of collision also by increasing the concentration. Okay, but yes, for here, yeah, I think it's in your book they have mentioned specifically that we need smaller amount. This is one of the good thing about enzyme, that they are active or they can work in at smaller amount because they can be reused. This is the explanation. Because they can be reused, they are free, they are not damaged, they are
Sir,
not destroyed. And there is always a limit of enzyme in your body. If there is destruction of enzyme due to any reason, due to disease process or due to infections and other stuff, so body would definitely replace that amount of enzyme. Okay, good. Okay, let's go to
sir,
part question number 2, part A.
sir, I have a question.
Yeah, okay, go ahead, Muhammad.
Sir, will the increase in amount of enzymes increase in amount need of energy?
Need of you mean energy? What? Sorry.
Yeah, like if there would be more enzyme, would we need more energy for like for the reaction?
Reaction is not necessarily the enzyme activity. Energy will remain. You probably need less time, to get the product. So the energy level probably is not that much affected. It's only by temperature or the you could increase or decrease. If you increase or decrease the temperature of the environment, you could also see yeah. But if you are increasing concentration, if you have larger amount and you are increasing the energy as well, so you have actually more enzyme that will work on the substrate very easily. The collision or interaction with substrate is very easy. So yes, this is absolutely possible. Yes.
Sir, I have
Yes,
a
Sabiha?
question. And enzymes are proteins, right? So, do our body just makes enzymes, or does it come from our food, the proteins we eat?
No, no, actually, this is a very good question. I think, yeah. So this is the question number 2 you're asking, right? To which group of chemical compound do enzymes belong, right? So,
Yes,
yeah. So first of all, yeah, let's talk about this. So what is the class chemical compound? The class of the
protein.
enzymes. Oh, yes, Sabiha?
Protein.
Yeah, they are proteins. So now coming to your question that do we need it? Our body definitely needs enzyme, but they are different from the proteins like egg protein. You have meat and plant sources protein like pulses. So in that case, this protein is different from. They are protein in nature. Chemically, they are related to protein, but proteins are of different kinds, right? So enzyme is one kind of protein, right? But enzyme definitely are also digested. That is why if you're taking some enzyme, they are not stable. They will be digested in the stomach or in the intestine, right? But enzymes are generated or synthesized inside the cell where they are needed. So they are cellular catalysts. They are needed where they are not digested or broken down. Definitely, they have a lifespan, of course, and that's why you have the resynthesis. If there is, we are using some drugs that will inhibit enzymes, right? Some chemicals to inhibit certain enzyme, like in case of hypertension, blood pressure. You want to block some enzyme there, and then to stop the activity. But yes, enzymes, the enzyme that we are talking about are different from the proteins we are eating, right? So enzymes are needed in very small amount. If you're eating meat, fish, for example, rich in protein, egg protein, they are different proteins and they are digested and they release amino acids, and that will be part of our body, which is needed for the body. So is it clear, Sabiha? You have any other question?
Yes, that's clear. I just had another question that enzymes, they our body just makes them, or they come from our food?
No, they are also like for example, plant cells do have enzyme, but what enzymes we are talking about in biological catalysts. They are endogenous. Endogenous means they are part of your body. They are synthesized inside the body. They are synthesized in the body. We are not getting all enzyme from outside. These enzymes are, you might get it like papaya. I mentioned proteolytic enzymes are there. So they can, of course, be used. Even if you take it from outside, it will work. But the enzyme that are working in the cell in our body, they are synthesized in our body. They are, we are call it endogenous. Endogenous mean a part, natural part of your body. They are not exogenous. Exogenous mean you are taking from outside, but you could, in some cases, enzyme could also be taken as a drug. So that is, we call it exogenously, and they are produced in the laboratories. Okay, is it clear?
Yes, thank you.
Oh, welcome. Okay, could you please read part B then? Who is going to read part B? Uh, okay, Omahil, could you please read part B, question 2, and answer that as well? Yes, Omahil. Your microphone is switched on, but we can't hear you. Okay, I think you have some problem with the microphone. I'm not sure. Somebody can hear Omahil? Or maybe,
No,
I
sir.
can't.
yeah, so we cannot hear. Oh, it's okay. Omahil, I think you need to use your built-in speaker of the device rather than your headphone. If you remove that, probably it should work. Okay, let's, Rafi, would you like to answer question 2, part B? Yes, Rafi?
Yes, which one?
Yeah, question 2, part B.
Part B. Which
Yes.
enzymes work on carbohydrates, protein, and fat? The enzymes which work on
Yeah, which enzyme will work on number 1? Carbohydrates.
number one, carbohydrates will work. On
Okay, good.
proteins, on proteins, protease will work. And on
Uh-huh.
fats, lipase.
Lipase, very good. Lipase will work on lipids or fats. Proteases will break down proteins, and carbohydrates like amylase, you have it breaking down starch into glucose. Very good. Okay. Name the product C. Who is going to read that? Yes, Sabiha? Part C?
Name the products that are formed in each case. Do I answer?
Yeah, please. Yeah, go ahead.
Carbohydrates break down the carbohydrates and it turns into sugar.
Okay, starch into sugar, sucrose. Okay.
Proteins, and they break down proteins break down the proteins break down proteins, and it will break down, and the product will be amino acids.
Amino
and
acids. Yes. Okay. Then third,
and for
fats.
will be the product will be fatty acids and glycerin.
Yeah, fatty acid and glycerol. Yeah, fatty acids. Basically, these are, we cannot use fats in like in this form if they are bigger molecules. So they must be broken down. So fats are broken down into fatty acids and proteins into amino acids. Amino acids can easily be absorbed. You remember, we'll talk about digestion, inshallah. So probably we'll cover part of it over there as well. Okay, now go to question number 3. I think Muhammad, would you like to read and answer question 3, part A?
Yes, sir.
Okay.
So I cannot see it.
You cannot see it? Question 3. Okay.
Yes,
Can you
sir.
see now?
Not. Yes. An enzyme has an active site on its surface on which the reaction takes place. Use the idea of an active site to explain why an enzyme is specific for a particular substrate.
Mm-hmm.
So because the structure of the active site and the substrate matches, so they react.
Very good. Yeah, key and lock model, right? Lock and key. So that's why it can lock, enzyme can lock the substrate over there. So that is very particular because very good. Okay, would you like to answer B as well?
So why
Yeah.
enzymes are denatured at high temperatures? So because as the temperature increases above a specific level, so the enzymes are damaged because of
Damage. So
temperature, high
what do we call
temperature,
it?
denatured, being
Denatured. So
denatured.
which part is denatured?
The active site.
The active site is also denatured. Yeah, that is the one reason that it cannot fit anymore, this substrate. Yes, very good. The substrate cannot attach easily after the active site shape is changed. Okay, great. Okay, who is going to answer C then? C, Aisha?
Because once substrate has been broken down, the active site is present for another substrate.
Excellent. Active site is free. It's available. It can attach another, because active site is not, you know, damaged or changed. It's remain the same. It remains the same, so it can be okay. Very good. Okay, let's go to part question number 4. So who is going to read this question? And also you need to analyze the situation. Okay. Yes, Muhammad, would you like to read the question and explain the experiment?
Okay, sir. Lipase is an enzyme that breaks down fats. We can use an indicator to follow this reaction. The indicator is red to start with but turns yellow when all the fat has been broken down. And then look at the results. At what temperature does lipase work best? Um,
Okay.
so maybe at 40, yeah, 40 Celsius.
So first of all, you need, before answer the question, you look at the table. This is very important that you first of all try to understand what they are asking. What are the information given to you? Okay. So here is the table. They have a test tubes. These test tube has got is maintained at different temperature. So you can see 5 test tubes are there, and they have all maintained at different temperature. And they are saying, what does it contain? It contains the uh, fats, right? So they are saying the original color for the fats would be red, but if they are broken down by the enzyme lipase, so the color would change from red to yellow. Okay, the color will change from red to yellow. So now look at these tube, original color all red, and final color, because you maintain it for maybe little time. So now using these information, the questions are, so okay, who is going to answer? Javeria, would you like to answer A?
Yes.
Okay.
So I think the answer is 40 degrees centigrade. And that's
Okay.
because the final
How, what makes you say that?
temperature.
So how you can conclude that? On what basis?
Uh, well, as the question states, the
Hmm.
start with yellow when all the fat has been broken down. So an enzyme would be good if it would break down all of the substrate.
And what is the indicator for that, that all the
So
substrate
it
is
works
broken down?
better at 40 degrees.
No, I mean, what is the indicator? How would you know that all substrate is broken down?
The color changed from red to
The
yellow.
color
Very,
changed from red to yellow.
very good. Yes, that is the main indicator. Okay. So you could say that at 40, look at all, where exactly is the yellow? So orange means still it's not all the product is created or produced. Rather, some fats are still there. That's why color is not yellow. Okay. If it is completely yellow, 40 degrees Celsius, you could say that that is the optimal temperature where enzyme has worked the most. And that's why we could see, I hope everybody can understand that. So what the point is that whenever you are asked such question in the exam, don't jump to the answer. Okay. Read the whole information. So the way Muhammad read the whole scenario, that lipase is an enzyme that breaks down fats. So we can use this indicator to follow this reaction. And they are saying the indicator is red to start. I mean, when the fat is there, it's red. But when it's converted or degraded or broken down, then the color will be yellow. So these information are given to you. And they are saying this reaction is maintained at different temperature. Okay. So first of all, Javeri has already answered at what temperature. So now look at temperature first. They are asking temperature. The enzyme works the best. Okay. So you would see which one has got yellow color. So this is the one, right? So that is the correct answer, that at 40, the temperature is 40 is optimal. Okay, Hania, would you go for part B?
Okay, sure.
Okay,
Why do you think the color did not change in test tube 1?
correct.
Um, I think because the reaction was carried out at 0 degrees Celsius,
Hmm.
um, so the movement of the molecules was low,
Hmm.
and the substrate and enzyme molecules would collide really.
Very good. This is very scientific explanation, Hania. Wonderful. Yes, it's 0 degrees Celsius, no energy, no movement. We call it dormant. The way it's not active. Enzyme is not active at 0 degrees Celsius. That is why red color is originally red. There is no product at all. Fat was not converted into fatty acids, right? So, okay, very good. Yes, that is the explanation. Okay, would you like to explain C as well?
Sure.
Okay.
Why do you think the color did not change in test tube 5? Well, I think it did not change because the reaction was carried out at 100 degrees Celsius.
Hmm.
So the enzyme was denatured at this higher temperature, and the fats were not broken down, so it remained
Correct.
red.
Yeah. So, what happened to the enzyme at high degrees Celsius? 100 degrees Celsius, sorry.
It denatured.
It denatured, yeah. Denatured means enzyme is now damaged, is not going to work. Okay, so let's see. Aisha, would you like to answer D?
Predict what you think would happen if you warmed up test tube 1 to 40 degrees Celsius and cooled down test tube 5 to 40 degrees Celsius. Try to
Hmm.
explain your prediction. If we warm test tube 1, then the reaction would take place, but if we cooled test tube 5, there would be no reaction because the enzyme has been denatured.
Wonderful, yeah. So they are now giving another scenario here in this experiment that how about if you will cool it down to 40 instead of 100? So do you think this is going to work? No, because the active site is once damaged is irreversible. So this damage is irreversible. It cannot be regained. This enzyme cannot regain the function. But what about one?
The reaction would take place.
The reaction will take place because the enzyme was not active because of the temperature, right? At 0 degrees Celsius. And enzyme is not damaged at a very low temperature. And most of the time, you might have seen the biologicals or enzymes that we are using for in industries or also in the market, in the hospital also. So we store them at freezing temperature, like in the freezers. So we have different freezing temperature, -4 or -60, -80, those enzymes that are highly sensitive to temperature, right? So whenever it's needed, we just warm them up and then it starts working. Very good. So the here what they want to know from you, if you understand denaturing is irreversible process, and the rest, yeah. And you could see here we are increasing the temperature from 10 to 40. You are also increasing the activity of the enzyme. So this is one of the things that you should keep in mind. Okay, let's go to uh, question 5. So let's see who is going to answer that. Somebody who is not part. Yeah, Umahil, if you can speak, I'm not sure, we can't hear you. You're most welcome to participate. Yeah, go ahead. Yes, Umahil.
Teacher, I mean, sir, maybe mic doesn't work, and can she write the answer in the chat?
What is not working? Sorry, you can't hear us. Can you hear us, Umahil?
Teacher, why can't she write it in the chat box? If she can't,
Chat
in
box is equal disabled because then we do receive a lot of interruption. So but I think what basically I would say, if somebody cannot hear, this is recorded, okay? So we'll upload this and you could still see the answers because we are discussing detailed explanation for the answers. So in the chat you will just write the answer. You could even find these answers anywhere in the in the key which is provided with the book. But the only thing which is important to understand those answers, why would we choose these answer, what is the explanation for that? Okay, so let's go to question 5. But if we still have some problem, yes, most welcome. We can chat in the group, WhatsApp group that we have, and I'm inshallah I will try to explain there as well if it's needed. But I think you can still go through the recording. Okay, question number 5. Who is going to read that?
Can I do it, Anya Basi?
Yeah, okay, go ahead.
The number of factors can alter the rate of an enzyme controlled reaction. Say what each of the following would do to the rate of reaction and give your reasons.
Yeah,
A.
explanation.
Increase
Yes.
in enzyme concentration. Uh, it will increase in an enzyme concentration because of the rate of the reaction, because more of the substrate molecules can fit into the active site.
Yeah, the more enzyme concentration is available, you probably can have more product formation because substrate will be easily interacting. Okay, yeah, that's true. Concentration will also increase.
Can I tell the next one?
Okay, okay. Okay, number B, part B. Yes, go ahead now.
A decrease in temperature. A decrease in temperature would decrease the rate of reaction because molecules become slower, and the temperature at the lower temperature, and they would collide less, so the substrate molecules would fit into the active site of the
Yeah,
enzyme.
collision. You could say when there is a decrease in temperature, you're also decreasing the moment or collision between a substrate and enzyme. Okay, so that collision is decreased. That is why you may not see product formation. So substrate will not fit in easily in low temperature. As you have seen in question 4, at 0, the enzyme is dormant. At 10 degrees Celsius, some enzyme is working, but part of it is not working. Still, it's not that active. Substrate and enzyme interaction collision is not that efficient. You are increasing the temperature, yes, that will. If you are decreasing temperature, you are decreasing the interaction between substrate and enzyme. Very good. Okay, C, part C, Muhammad. Yes, Muhammad, you can hear us. Or Sabiha, would you like to answer? Yes.
Yes, sir. Can I answer, Sabiha?
Yeah, go ahead, Sabiha.
Lowering in pH and the if the pH, the amount of pH is like it's decreasing, then the enzyme will go lower.
So how would affect pH? How would it affect enzyme activity, the pH? You're saying that if pH will be lower, the enzyme activity will also be lowered?
Yes.
What did we discuss? What did we discuss about the effect on pH? Impact of pH on enzyme activity. Did we say that if decreased pH, enzyme activity will be decreased? Anybody would like to comment on that? Muhammad, would you like to add something? Or Ahmad? Okay, Muhammad,
So
would
I'm
you
not
like to
sure.
comment?
So I'm not sure.
Okay, lowering of the pH. What was the effect of pH? Hania?
Yes, sir.
Oh, yeah. So what do you think? How pH will affect the enzyme activity? A lowering of pH, part C, question 5.
Because it would decrease the rate of the reaction, because the active site of the enzyme uh, would change at a lower pH, and the substrate molecules would not be able to fit into the altered active site of an enzyme molecule.
But they did not mention the nature of enzyme. We don't know if at what pH this enzyme is active, right? So in a pH, you would say that it depends on the nature of the enzyme. Some enzymes are active at lower pH, some enzymes at neutral, some are active at higher pH, right? At basic and alkaline environment, some are active in acidic environment. So lowering the pH may decrease or increase, depending on the nature of the enzyme. Those enzymes that are active in acidic environment, so if you're lowering the pH, you're probably increasing the activity of the enzyme, right? But those enzymes that are working at higher pH, which is alkaline, if you decrease the pH, definitely you are also, so it is, if you're decreasing pH, you are also decreasing enzyme activity, because enzyme needs, as I mentioned before in this chapter, one of the, I think, experiment on proteins, proteases, that they are working in small intestine, and that is a little working at higher pH. If you lower the pH, so it will definitely decrease the activity of those enzymes that are working in higher pH. So decreasing pH or lowering pH, okay, and any reduction in pH would definitely decrease enzyme activity. But yes, for those enzymes that are working in low pH, so their activity will not be affected. They will not be inactive because they are working in low pH, right? So it depends on the nature of enzyme. Is it okay? Anybody would like to add something, or if you are not clear, let me know, please. Then we'll move further to next part, next question.
No.
Okay. Yes. So let me give you.
Sir, what
So you
happens if,
not share.
sir,
Yeah, I will
if,
share with you another picture, I think.
sir, and sir, if the pH of the enzyme is neutral, then will the lowering of the pH affect it?
You're very good. Yeah, this is a very good question. Again, if it is neutral and you are lowering the pH, yes, that will affect the activity. Very good. Excellent. Yes, that's true. Because you should keep in mind what enzymes are active at what pH. So if they are active at neutral, so even in lowering the pH or increasing the pH would both affect the activity of enzyme. But if enzyme is active in low pH, lowering the pH is not going to affect much on the activity of enzyme. Okay. So, okay, very good. So let's talk about question 6. What do you think? Who is going to read this and explain it as well? Okay, let me see who is not participated. Those who have joined, I think now somebody would like to. Yes, Mariam, would you like to discuss question 6, please?
Yes.
Oh, go ahead. Okay, please.
Question 6. The following experiment was set up to investigate the action of protease on egg white. When enzymes break down the protein in egg white, it changes from cloudy to clear. A.
Okay.
Explain why contents of test tube A went clear after 13 minutes.
So now look, first of all, you have read this scenario. So now the second part after you have read this part is to look at the tubes. Tube A and tube B. So what is given over there? What's 1 cm cube or 1 ml water, 1 ml or 1 cm cube of pepsin, and 5 cm cube of egg white. Okay, and there are three things in tube A. So pepsin is a proteolytic enzyme, right? Then why not? Why egg? Can we put here starch, or starch will also work?
No, it will not work.
Do you think we can also put starch here?
No,
Carbohydrates here?
no,
Why
No.
egg protein?
Say
because
because then we would
pepsin.
need carbohydrates.
Yeah, so egg white has got protein. That is why pepsin is a proteolytic enzyme. Okay, so now you explain the question. In the part in tube B, what they are saying, only 2 cm cube water and 5 cm cube egg white. So what is the difference in both these tube A and B? What is lacking in tube B?
In
Tube.
test tube B, there isn't pepsin.
Yeah, the pepsin is not there. Okay, now explain question A. Part A, explain why the contents of test tube A went clear after 15 minutes.
Well, inside test tube A, that there's the enzyme pepsin, so it broke down all the egg white into amino acids. But inside test tube B,
But why the solution is clear then?
the solution is clear because the enzyme because the protein is broken down.
Okay, then. If protein is not
And
broken down, like in case of tube B, it's turbid, right? It's not clear.
Can I tell?
Yeah, okay.
The pepsin enzyme broke down the white protein into amino acids. That's why the contents
So amino acid, they are soluble in water. Okay, that's why this is one of the thing. Yeah, Mariam already answered that it's broken down by the pepsin. That's why it has got an enzyme, right? So tube A, after 15 minutes, the egg white is broken down, protein is broken down, and it's clear now. While in case of egg white, if you put egg white a little bit in water, you see it's frothy. Well, you would see even the solution is very turbid. It's not clear. It's cloudy. Why is cloudy? Because this protein is not making frothy. The solution is frothy. Okay, and there's one of the things that you should keep in mind, that enzyme is needed to break down a protein into amino acids, and amino acid then will give clear, so it will not make froth, or so the solution will be very clear. Okay, part B. Now, who is going to explain part B? Rafe, would you like to explain part B?
Okay. Explain why the content of test tube B stayed cloudy after 15 minutes. Test tube B didn't contain enzyme pepsin, so egg white protein was not broken down and content stayed cloudy.
Yes, that's true. Excellent, Rafi. Very good. Yeah, since the enzyme is not there, even if you keep it the whole day, it's not going to change because you need enzyme here. So test tube B would not be clear. It will be cloudy. As I mentioned that if you stir it, even if you add a drop of egg white to water, any test tube with water, you would see the solution would be very turbid or cloudy because it's not soluble. It's giving frothy or cloudy look to the solution. But when it is broken down, no protein is there, so there are amino acids that are they are making clear solution. Okay, how could you increase the speed of the reactions? Part C. Okay, part C, iPad. It's better if you would like to mention your name, but go ahead. Yes, please. Part C.
How could you increase the span of the reaction in test tube A?
Okay.
By increasing the temperature, by
Yes.
increasing the concentration of enzyme pepsin,
Well, yes.
by lowering the pH, by adding dilute hydrochloric acid.
Wow. Wonderful. Excellent. You have mentioned more than what is needed, even, right? Yeah, she's saying that temperature alone would definitely speed up the reaction, and she already integrating those concepts that concentration of enzyme would give more chance for the substrate to interact and collide and work on it. And yes, this is what she said. Proteolytic enzymes are, as we mentioned before, proteases are very active on alkaline, right, are basic. But pepsin is also an acidic environment. If you are making the environment acidic, probably maybe breaking down the proteins. But some of the proteases that we mentioned in case of in the intestine, where they work in alkaline nature. So pH effect should not be changed because it might affect the rate of reaction. It might slow down if the enzyme is activated alkaline pH. But yes, temperature concentration is going to speed up the reaction. That's true. pH has got variable effect. It depends on the nature of enzyme. But pepsin is also available in the stomach. So in acidic environment, probably should work faster than alkaline environment. Okay. What about part D? Who is going to read? Javeria.
Sabiha?
Sabiha, you want to comment or?
I'm asking, can I do D part?
Okay, can we give chance to Javeria, please? D, and there are more questions, then we'll give you chance. Don't worry.
Okay.
Yes, Javeria, beta. You are with us? Part D?
What do you think the egg white protein was broken down to form?
Yes.
The part B is which enzyme work best in acid conditions.
No, D. D. She's reading D. Part D. Javeria is reading part D.
Of
Yes.
Maybe a type of amino acids.
What do you think the egg white proteins was broken down to form? So what is the product here? Protein, egg white is a substrate, right? Pepsin was there. So what is going to be formed?
Maybe a type of amino.
Specific question, answer. I think we discussed about if protein is broken down, what is the product then? Umahil?
Can you hear me now?
Yeah, we can hear you. Yes, Umahil.
Amino acids.
Yes, very good. Amino acids, right? Javeri, you got it? You're right that they're broken down. Of course, protein is broken down to a smaller unit, and that is amino acids. Like fats are broken down to fatty acids, carbohydrate into glucose, starch into glucose. So proteins are broken down into amino acids because proteins are made of amino acids that are joined together. Okay, so yes, great. Right, let's go to question 7 because we are running short of time. We need to finish this end chapter question. Okay, go ahead. Question 7, who is going to read? Aisha, would you like to read?
The graph shows how temperature affects an enzyme controlled reaction. A. Describe what is happening between X and Y. The,
Explain what is happening.
the enzyme is breaking down the substrate,
So what do you call it? The enzyme activity is increased or decreased?
increasing.
Very good. Enzyme. So between X and Y, you would say the enzyme activity is increased because you would see rate of reaction. So look at the graph, what they have. So answer occur. Your answer is absolutely correct. You still can say the product is, but here they have mentioned rate of reaction and temperature. So what is going to happen between X and Y? If you go from X and Y, you could see temperature is increased. The increase in temperature is increasing rate of reaction. This is the exact answer. Okay, you could answer in many way, you want, you could have further explanation, but try to be focused on what they are asking, what factors, what information they have given you in the graph. They are talking about rate of reaction and temperature. So what is going to happen between X and Y? Then increase in temperature gradually is increasing rate of reaction. That's why straight line. Okay, very good. Okay, let's uh, yes, Sabiha, would you like to answer part 2?
Of part A.
Part A, yes, number 2.
Explain what is happening between Y and Z. Between Y and Z, the temperature is increasing more, and so the enzyme is denaturing.
Yeah, so if you would like to answer according to the graph, what would you write first? This explanation is correct, absolutely correct. But the point is, you still did not answer the question. They're saying, what is happening? If you would like to explain somebody, they have not given you anything, any product, any substrate. So nobody would understand if what you know the basic background. That's why you can explain it. But in terms of this graph, how would you answer this question? What is happening between Y and Z? Yes, Sabiha, yes, go ahead.
Temperature is increasing like over the optimum temperature, the 40 Celsius. So then what is after over 40 degrees Celsius? So then what is happening that it's going to 50 degrees Celsius Z, and that's why the enzyme, the active site is changing the shape of it
Yeah,
in the
but
substrate and
they are saying what is
more.
happening, right? So that is that this is actually detailed explanation. So we are asking what is happening between Y and Z. As I mentioned, between Y and Z, with the increase in temperature, the rate of reaction is decreasing. Okay, so when rate of reaction is this is the answer, and then you explain because enzyme is denatured. There was optimal temperature where enzyme activity was the most at 35 or around 30, yeah, around 35, and then you further increase the temperature, so the active site is damaged or enzyme is denatured. But if you want to answer this question, first of all, you must write that what's going on between Y and Z is that increase in temperature, further increase in temperature is decreasing the rate of reaction. The rate of reaction is decreased. So why this is decreased, you can write it down. If you have time, you can write it down. Yes, because enzyme is denatured. Okay, B, part B. Omahil, yes,
Yes.
yes, go ahead. Part B, suggest how increasing temperature affects the rate of reaction of the enzyme.
Initially, enzyme reaction rate increases with an increase in temperature as molecules move faster and collide more with each other, and it will reach at its peak at optimum temperature. With, with further increase in the temperature, the rate of reaction decreases rapidly because enzyme gets denatured.
Very good. Yes, that's very good. Yeah, this is the correct answer. You're right. So basically, they want to know that What happens to this? Okay, great. So enzyme is denatured. Yes, when you are increasing temperature up to certain level, you are increasing thermal energy. Moment of the molecule of enzyme and a substrate can easily collide and they can fit in, and then product will be, rate of reaction will be increased, okay, by increasing temperature, but up to optimal temperature. There's always a limit for temperature. Okay, what about part C, Javeria? Yes, Javeria, part C.
The active, what's happening to the active site of the enzyme at the higher temperatures?
Mhm.
The active site is denaturing at higher temperatures above 40 degrees.
Correct. Yeah, that's very good. A high temperature is denatured. The active site is no more functional because it's already damaged. Okay.
Can I do the next question?
Yeah, go ahead. Go ahead, please.
Uh,
Please.
is it D?
This graph shows, yeah, I think, yeah, they're
Mm,
asking question. This graph is showing the pH effect on the rate of two enzymes controlled reactions. Yes, go ahead, Rafi.
which enzyme works best in acid conditions?
So there,
A,
you first look at the graph. What they have given you, rate of reaction,
enzyme
and then
A
the
and
pH.
enzyme B.
So what enzyme is working best at acidic condition? A or B?
Enzyme A is working best in acidic
Enzyme
conditions
A. Why? Because the pH is,
because pH is low, or because it's on the right side, it's on 2.
yeah, below
Not
neutral. Okay, neutral point is 7. Below 7 is acidic environment. Above 7 is
it's
alkaline environment. Okay, yes, that's very good. That's why enzyme is A is giving you this peak. This peak is showing optimal activity. See here? So what does it mean? If we are increasing the pH up to a certain level, you could see here, the enzyme activity is also increasing. So, but if you are further increasing, so this enzyme is very active at this pH range. It has got a range of pH. That's what you mentioned before. If we lowering the pH or decreasing the pH will affect enzyme or not. So in this case, here if you lower the pH, the enzyme activity will be increased because this enzyme is active in acidic environment. Okay, very good. What about B? Is active in what part?
um which enzyme B works at an alkali question.
In alkaline, yeah. So if you lower the pH for this enzyme, do you think the rate of reaction will be increased?
The
If you lower the pH for B?
no, because the
Yes.
pH.
So would you please explain that? We can't hear you.
No, it will not.
Why? Yes, Sabia?
Yes, I should
Yeah,
answer.
but for B, we are asking a question that is active at alkaline environment. If you lower the pH for this B, enzyme B, the rate of reaction will be increase or decrease?
It will decrease
It will decrease. Why?
because if you're gonna lower it, it's gonna turn to acidic, and um then out of acidic, it won't be able to work.
Very good. Exactly. This is absolutely true. That B is active C above 7. It's not working below 7. If you're lowering the pH, you are basically bringing it down to the acidic, so it's not going to work. So I will just quickly finish this and then because enough prayer time. So what does the graph tell you about the range of pH over which each enzyme is working or is active? Yes, Javeria?
Um, it tells us that enzyme A works in highly acidic in highly acidic
If you want to
environment,
see the range,
and
so where would you give some value? Would you like to say which value do you think would be more appropriate for this enzyme to work?
um,
Which range?
um, pH 2.
Range mean there is a range between 0 till 4, for example. So which range is very good for this enzyme to work?
2.
This is not range. This is one value. I'm saying range. Range mean value
Uh,
from
one
0 till 4. So you have to say this range is very good for the enzyme.
1 till 3 is the best range for this enzyme.
1 to 3. Oh, really?
Well, it only works in these three
Yes,
pH.
Aisha?
It's 2.
So they're asking range, okay? So when they're asking range, you need to focus on the enzyme reaction, where exactly it's increasing. It start from 0, you don't have any rate of reaction. Around 1 is going to increase, right? So what's going to happen to this enzyme, and where exactly is the maximum and what pH, and then where exactly is declining, decreasing?
It's at its peak at 2,
Mhm.
and then it starts to denature.
It start to denature. Very good. So starting from 1, it's increasing 1.2, 1.3 till 2, which is maximum here. This is the optimal pH where the enzyme is very active. If you further it, you still have enzyme reaction, but it's declining gradually. You could see here a part of it is declining, right? Can you see my cursor?
Yes.
Okay,
Yes.
so
Yes.
you could see here, see it's decreasing on the other side, on the right side. The left side is increasing up to the top. So from here, if you still want, uh, we would say that, let's suppose if you want 50%, so anything below 3, from 1 till 2 point, you could say 7 probably is the best. Otherwise, 3 is not free, because you could see here again, very low. Rate of reaction is very slow. Although enzyme is still working, but the efficacy or efficiency of enzyme is very low if you are so the range of pH is from starting from somehow 1.2 till 2.8, you could say, where 50% activity will still be there. 50% is the half of the enzyme activity, but 3 is of course not. Anything going beyond 3 is not good for this enzyme. Same is the case here. For it's just above 7 and below 9 point, you could see below 9, okay, then it's going to decrease. So for B enzyme, this is the range starting from 1 till 2.8 and starting from 7.2 till 8.5. So this is the range where these two enzymes are working. Is it clear? So the next question is regarding the industrial use of enzyme. We did not, we have not covered that part in the chapter. We'll see if you want me to cover that industrial part. It's a little bit complicated maybe for you, because you don't have background knowledge about things. So I'm not sure, but if you still want it, we can cover it. But you can also see if that is needed for the exam, or you want to study in grade 8 or 9. I think this will be
Sir,
probably there.
we haven't, sir, we haven't done this.
Yes, this was omit.
Oh, this is omitted. Okay,
Was
so we can,
omitted.
we'll see if we have time. We can. Okay, thank you so much. I think we'll upload this inshallah on the YouTube channel. You could just see if you have any question. You can ask me in the chat, inshallah. Now it's a prayer time. I can take quick question if you have one or two questions. Do you have any question, any query?
No.
Okay, see you then, inshallah. Assalamu alaikum wa rahmatullahi wa barakatuh.
Walaikum assalam wa rahmatullahi wa barakatuh.
Yes.
Yeah, man.